Hello students, today we'll discuss about the important topic that is called as methods of gene transfer. In this topic, we'll be studying how the genes are transferred, especially in case of plants or say some other microorganisms, in order to generate the recombinant cells or recombinant organisms. In this case, basically the methods of biotechnology or recombinant DNA technology are used where the desired gene is inserted or transferred into the desired organisms. It might be in animals or in plants or in organisms. So, in this aspect, mainly we will be discussing what are the methods that are transferred for the DNA into the other organisms. So coming to the methods of DNA transfer, there are two methods usually followed. The first method is the direct transfer of DNA, which is called as vectorless transfer of gene or the DNA. Vectorless transfer means in this case no mediator is required or no mediator or without the help of an transforming vehicle, directly the gene or the DNA is made to come in contact with the host cell or the recipient cell, and directly without the help of the vector or without the help of the mediator, the DNA is transferred in case of direct DNA transfer. Whereas in the second method, that is indirect DNA transfer, which is also called as vector mediated DNA transfer. In this case, a mediator or vector is used to transfer the DNA, so it is called as vector mediated DNA transfer. These are the two methods of DNA transfer. So coming to the first method, that is direct gene transfer, which doesn't require any mediator or any vector for transferring the gene. Direct gene transfer can be done by using different methods such as particle bombardment method, which is done by using an instrument called as gene gun, or which is also called as biolistic gun, or this method is also called as ballistic method. The second method is called as liposome mediated gene transfer. Third method is electroporation of protoplast. Next one is microinjection. The last one which we are studying is the silicon carbide fibers. So mainly by using these 5 important methods, gene is directly transferred into the recipient or the host cell. Whereas coming to the second method, that is indirect method of DNA transfer, which is also called as vector based method, where we are using an mediator to carry our gene and to transfer that into the desired host or recipient cell. So in vector based method, mainly the microorganisms such as bacteria or viruses are used for transferring the gene, and these act as the transforming vehicles. The type of the bacterium which is usually, especially in case of plant transformation, where we want to transfer the gene from one organism into plants, means if you want to create a genetically modified crop, at that case mainly for the plant transformation purpose, the vector or the bacterium used is the bacterium called as agrobacterium. Usually 2 species of agrobacterium are used. Those are Agrobacterium tumefaciens and Agrobacterium rhizogens. So by using these 2 bacteria, the genes are transferred into the plants. And the second type of the vehicle or the vector used for the transformation purpose is the viruses, and those are called as the viral vectors. Example are cauliflower mosaic virus, which we call it as CAMV, and the second one which is popularly used is called as the Gemini virus, which we will be discussing at the later steps. And coming to the direct DNA transfer, which is also called as vectorless. Again, this direct DNA transfer, it can be done in 2 ways or 2 methods. First one is physical gene transfer methods, second one is chemical gene transfer methods. As you all know, direct DNA transfer method means, in this case, the host or the recipient cells will come in direct contact with the naked DNA or the naked gene. We are not at all going to insert this DNA into any vectors, or we are not at all going to ligate the desired gene into any other vectors. Instead, in this case, what happens, the desired gene or the required DNA directly comes in contact with the host or the recipient cell, which could be done in two methods. First one is the physical method of gene transfer, second one is the chemical method of gene transfer. As the name indicates, physical method of gene transfer means here we use some physical instruments in order to penetrate the DNA into the recipient cell, and those will be discussing in the physical gene transfer methods. Whereas in the second method, that is chemical gene transfer methods, in this case what happens, in order to aid or in order to accelerate the entry of the desired DNA or desired gene into the host or the recipient cell, we use some chemicals for the transformation purpose. Those we will be discussing in chemical gene transfer methods. So coming to the first method which we have to study in physical gene transfer method is the particle bombardment method, or which is also called as micro projectile method. And there are different names for this method, could be called as particle bombardment method, biolistic method, particle gun, gene gun, or which is also called as what, bioblaster. In this case, what is done is, and it is also said that this method is the most effective method of gene transfer, especially in order to create the transgenic plants. And in this method, or this method was developed by Professor Stanford and his co-workers at Cornell University, USA in the year 1987. And as the name itself indicates, biogun, or we call it as what, gene gun or bioblaster. In this case, what happens, or this bioblaster or particle gun, it shoots the desired DNA or foreign DNA into plant cells or tissues at a very high speed. That is why it is called as what, the gene gun method or the particle gun method. And in order to transfer the desired gene, especially some of the molecules are used here, which we call them as what, microcarriers or micro projectiles. So in this slide you can find, we are using, so here I have written micro projectiles or micro carriers, which are nothing but small particles which are made up of tungsten or gold particles. And these tungsten or gold particles, you can find here the particles which are made up of gold and tungsten. In this case, these particles are coated with the desired DNA which you have isolated or which you want to transfer into the recipient cell, or say for example in the plant. If you want to transfer it into the plant, the desired gene, then what we need to do is we need to coat the tungsten particles or the gold particles, which we are going to call them as what, the micro carriers, and these are inserted in the gene gun. And then with a high pressure or with a high velocity, these are pushed downward so that they can rupture the disc which is placed below this instrument. In the later slide, how it is done, you are going to find out. So with a high velocity, whatever the gold and tungsten particles which are coated with the desired DNA are there, they are shooted or bombarded against the desired tissue where you want to transfer your desired gene, so that the recipient cell or the recipient host can receive our DNA very easily. So this is what actually done in case of the gene gun method or the biolistic method or the particle bombardment method. So in the later stage or in the later slide, you can find that other methods are also there, which we are going to study. So you can find here, which I have read. There are other names for this technique: particle gun, gene gun, bioblaster. The DNA is coated with tungsten gold particles, and these tungsten or gold particles are called as microcarriers, and these microcarriers are inserted in the projectiles, which are called as the macrocarriers. They are inserted in the apparatus, pushed down by rupturing the disc. So this is all what I have discussed. So clearly in this slide you can find how actually the gene gun works. Actually, to operate this instrument, the gas which we use here is the gas acceleration tube, which is given. So this one is connected to the helium gas cylinders. So that gas is passed into this in order to create a, that is, with a high velocity the gas is passed into this, which is going to rupture the disc, and then within this you can find the microcarriers, that is gold and tungsten particles coated with the desired DNA or the desired gene, and below you can find. So this is whole instrument, we are going to call it as what, the gene gun. So upper side we are going to insert a high pressure or high velocity with the help of the helium gas, and here mainly the tungsten or gold particles, which we call them as microcarriers, are placed. Then with a high velocity, whatever the tungsten and gold particles are there, they are shooted or bombarded against the desired plant or the desired host tissue, which is going to rupture or which is going to pass across the cell wall or the cell membrane of the host or the tissue, and later it is going to be inserted inside the host or recipient cell in order to make the cell as a transformant. So this is what actually done in case of the gene gun. So this is the conventional gene gun, how actually it works. That is, in the previous slide you found that is just the diagrammatic representation, and this is how actually the conventional gene gun is there. So these are the cylinders which consists of the helium gas. This is the pipeline through which the helium gas is inserted, and here you are going to find the microparticles. So with a high velocity, so below here kept the microplate on which you can place the embryonic cells or the callus or any tissue into which you want to insert your desired gene. So with a high velocity they are bombarded against the tissue which is kept below this, so that the tungsten and gold particles can break the cell wall of the tissue, which can invade or enter into the desired host or the recipient cell. Another type of the gene gun, which is called as handy gene gun, is called as the Helios gene gun method, which exactly appears like the gun within which we are going to load the bullets. So normal gun, usually what is done, the bullets are loaded, but in this case whatever the golden and tungsten particles are there, they act like the bullets. So this gene gun is actually loaded with the microparticles made up of tungsten or the gold, which are coated with the desired DNA which you wanted to insert into the desired host or the recipient cell. With a low pressure helium pulse, it is going to deliver the coated gold particles into the any target cell or the tissue. So you can find here gene delivery through the Helios gene gun method. So on the Petri plate, the tissue or the plant into which you want to transfer your gene is kept, and across or against the leaf, by using the gene gun, the tungsten and gold particles coated with the desired gene are shooted against the plant tissue into which the gene can be inserted. So this is actually what how the particle bombardment or the ballistic or the particle gene gun method is done. And the next method is called as the electroporation method. So in the electroporation method, this is also very important method where we can easily transfer our naked DNA or the desired DNA into the plant. In electroporation, actually what is done, the low voltage or say somehow slightly high voltage electrical impulses are used, or an high electric short is created in order to disturb the cell membrane of the protoplast, so that our DNA can be easily inserted into the desired host or the recipient cell. So this method, electro stands for what? Electric shock, or a kind of current or a voltage, electrical voltage is used. Poration stands for just to loosen the cell membrane or just to form the pores on the cell membrane, so that through these pores our desired DNA can be entered inside the protoplast, so that the cell can receive our desired gene or the desired DNA. So this is how actually the electroporation cell process occurs: before applying the electric pulse, during electric pulse, and after the pulse you can find. So in this slide you can find that actually how it is done. Here is the plant cell and here is the protoplast. The difference between the cell and the protoplast is that cell wall is removed. When we remove the cell wall, if only the cell is left with the cell membrane, then it will become what? The protoplast. And here is the DNA or the gene which you want to transfer it into the plant cell. So now we want to transfer this DNA into this protoplast. To do that, what? In an cuvette or in a tube, both the protoplast, that is the plant cell into which you want to transfer your DNA, is taken, and also the desired gene which you want to take, they are taken, and then the power, that is electric field, is applied. So when the electric field is applied, as you can find in the previous slide, so when you apply the electric field, here you can find the cell membrane is intact, but when you apply the electric field, what happens? You can find the formation of the pores over here. So why the pores are formed here? You know very well, the cell membrane is mainly made up of what? The lipids, and that too we call it as what? The bilipid layer. Cell membrane is made up of bilipid layer. So when we apply the electric field to this cell membrane, disturbance in the cell membrane, or what happens, rearrangement of the lipids that are found in the cell membrane occurs. So due to this rearrangement of the lipids that are found across the cell membrane, what happens? There is a disturbance in the distribution or the arrangement of the lipid, which leads to the formation of the pores on the cell membrane. So like this, the intact cell membrane becomes porous. Through this porous cell membrane, now the DNA can be, or the genes can be easily entered into the protoplast. Now you can call this cell as what? The recombinant cell, or you can call it as what? The transformant which has received our desired gene. Later, as usual, with the help of the normal mechanism, the healing of the cell membrane occurs. Healing of the cell membrane means whatever the lipids which were present in the cell membrane were there, they are going to rearrange themselves, or healing of the cell membrane occurs, again the cell membrane becomes intact. So like this, what happens? The DNA can be made very easily to penetrate across the porous cell membrane in order to create a competent cell, or in order to create an transformed cell. So this is what the electroporation technique. It is very simple, so which basically involves the use of high field strength electrical impulses to permeabilize, or which allows the cell membrane for the uptake of DNA. Especially this technique is used to deliver the DNA into intact plant cells and the protoplast. And electroporation has been successfully used for the production of transgenic plants, especially in cereals such as rice, wheat, etc. So this is regarding the electroporation technique. The next method, that is which we need to study in the physical method of gene transfer, is the micro injection. So the name itself indicates what is this micro injection. In this case, what is done? The micropipette, which is made up of very fine glass, is used to inject the gene at microscopic level, and this technique is called as the micro injection. Micro injection usually involves the direct mechanical introduction of the DNA into the nucleus or the cytoplasm. So in this case actually what is done, the micropipettes, or I can say microcapillary injection pipettes, are used to insert the DNA directly into the nucleus or the cytoplasm where the DNA is located. In order to do this, the size of the micropipette you can find here, 0.5 to 10 micrometer diameter tips are used to transfer the DNA into the cytoplasm or the nucleus of the recipient cell. You can find here which are the usually here what happens in this technique, the traditional compound microscope of around 200x magnification, or the dissecting stereo microscope of around 40 to 50x microscopes are used. Under the microscope what is done, you can find in this slide, especially here in this case. Under the microscope, target cell is positioned and cell membrane and nuclear envelope are penetrated with the help of micropipettes or the syringes. This is how actually by using these. So here in this you can find the needle or the micro injectors through which the gene is inserted into the desired host or the cell. And actually how this micro injection is done, in order to do this, protoplasts are taken, and those protoplasts are, so I am going to call it as what, the protoplast. You know very well, protoplast means it is the, say for example, a plant cell where the cell membrane is removed, only the cell membrane is kept for the cell. These protoplasts are immobilized in low melting agar while working under microscope. So we need to place the cells in the next slide. You will come to know how actually they are placed. We need to use an agar plate or the agar medium where the protoplast is immobilized. Immobilized means it is fixed in a particular position in the low melting agar medium while working under a microscope, and then with the help of a holding pipette and suction force it must be immobilized. So this is what the holding pipette. With the help of the holding pipette, the protoplast is held and it is inserted in the low melting agar medium, and later DNA is then directly injected into, so this is what the micro injection with a syringe. Directly with the help of the pipette, the gene is inserted into the cytoplasm or the nucleus. The injected cells are then cultured in vitro and regenerated into plant. So whatever these injected cells are there, you can find with the help of the micropipette or the needle directly into the nucleus, the gene or the desired DNA is inserted, and now these cells are called as what, the transformed cells, and these transformed cells are transferred into the say tissue culture medium where they are generated in an in vitro condition through the tissue culture technique. Like this we can create the transformed cells with the help of the micro injection. So here you clearly under the microscope how actually the micro injection, micro pipette, holder, etc. appears, that you can find in this slide. So this is the needle, this is the protoplast, this is the holding pipette. So this is all the low melting agar medium where the protoplast is inserted. You can find it is inserted, and with the help of the needle the DNA is inserted into the nucleus, and thus the protoplast is said to be transformed. The next method is liposome mediated transformation. This is the fourth method of transferring the gene into the recipient cell, or say for example in any tissue. Now the question comes, what are these liposomes? This liposome mediated transformation is also called as liposuction. Usually this method is used to deliver the required gene into the cell, or sometimes to deliver the drugs into the target cell. this liposome mediated transformation or liposuction is used. Now the question comes, what are these liposomes? Liposomes are microscopic and they are said to be very small sized bags which are made up of what? Lipids. The membrane of these lipid bags or the boundary of this lipid bags is mainly made up of phospholipid bilayers, and inside this vesicle or the bags they contain the chamber which is made up of water soluble molecules such as plasmids or foreign DNA. In the next slide you can find here. So these are what outside you can find. So these small, so what I am drawing here, so you consider these as what? The liposomes. So what are liposomes means? You are going to say these are the bags which are mainly made up of what? Lipids. That is, this boundary of the lipids is mainly made up of lipids. And what inside these liposomes, what is present or what can be inserted? Actually I can say now these liposomes are hollow, which doesn't contain anything, but inside these bags or inside these liposomes you can insert the desired gene or insert the desired DNA which you want to transfer into the recipient cell. That is what I said. Liposomes contain aqueous chamber which may contain water soluble molecules such as plasmids or foreign DNA. You can place your DNA, that is, say for example, take the DNA or take the gene which you want to transfer and place that DNA into a bag. What is the name of the bag or the bag where you want to insert your DNA? That bag is called as what? The liposome. What is the speciality of that liposome? Liposomes are mainly made up of lipids. Mainly the membrane is made up of lipids. Now the question comes, why only the liposomes, or why the liposomes must be made up of phospholipid bilayer? It is because now we want to transfer our desired DNA or gene into the another plant, another cell, and say for example protoplast, and the boundary of the protoplast or the membrane of the protoplast, you know very well, it may be an animal cell or maybe an plant cell having only the plasma membrane. Especially it is made up of what? The lipid only. Through fluid mosaic model we know that especially the cell membrane is, what is the composition of the cell membrane? It is made up of what? The lipids. Bilipid layer is present within which intrinsic proteins, extrinsic proteins, etc. are found, which we all studied. That is the reason why, that is mainly the composition of the cell membrane of the cell as well as the composition of the membrane of the liposomes, both must be same in order to deliver our desired gene or desired DNA into the host. That is why we call them as what? The liposomes. So in this case, what is done? In an coat or in a tube, we need to take the protoplast, that is the cell into which you want to transfer your DNA, and then you need to insert your liposomes. You need to insert your liposomes. So in this slide you can find, so these are the liposomes. Liposomes means I can say empty bags which are made up of lipids, and here is the your DNA which you want to transfer this into this cell. So this is the cell where you want to transfer your desired DNA. So next what happens, you just transfer this DNA into liposomes, and your this complex which is made up of liposome and DNA, we are going to call it as what? The lipoflex. Next what happens when you place this lipoplex complex, that is liposomes containing your DNA or the gene, take it into an test tube. So this is the test tube. So I am going to tell you here how you are going to do this. This is a test tube. In the test tube you have taken liposomes with your desired DNA. In the liposome the desired gene is there, and take this cell where you want to transfer your gene into the same test tube. Then what happens, first these liposomes go and bind themselves to the membrane of the protoplast where you want to transfer. So when it gets attached exactly at the point of attachment here, because The membrane of the liposome is also made up of lipids, whereas the membrane of this cell is also made up of what? Lipids. At the point of the contact here, what happens? Dissolution of the membrane occurs. The membrane gets dissolved, and whatever the content, actually what you have placed inside the liposomes, DNA is placed. So the content which is present inside the liposome can be easily taken inside the cell. Actually here what happens, the important method that occurs here, the principle which occurs here is endocytosis. Endocytosis, you know phagocytosis, in the same way endocytosis. Inside the cell, the liposomes are taken and the liposomes are digested, and where you can find a liposomal degradation, and our DNA is released. Later that DNA goes and integrates with the DNA of the host. So this is how easily, with the help of the liposomes, our DNA can be easily inserted into the desired host or the desired recipients. And in order to accelerate this, or the efficiency of the transformation in this case can be increased with the help of one chemical called as PEG, which we call it as polyethylene glycol. In the next latest chemical methods, there we are studying why we need to use this polyethylene glycol and all. So this is all about the fourth method, that is liposome mediated transformation or the liposuction. And the last method of gene transfer or the DNA transfer is the silicon carbide mediated transformation. So in this silicon carbide method, in this case what is done, the fibers which are made up of silicon are used, and the size of this fibers is about 0.3 to 0.6 pm in diameter, and about 10 to 100 pm in length. These fibers are, the purpose of using these fibers is they are capable of penetrating the cell wall and the plasma membrane, so that they can easily deliver the DNA into the cells. So in the earlier slide what you studied, to deliver the DNA we used the bags which are made up of the lipids. That means usually we made use of lipids in order to deliver the DNA. But in this case what is done, we use the silicon carbides which have very high affinity towards the cell membrane and the cell wall, so that our DNA can be inserted into the cells. And actually what is done here, you can find here in an container or in a test tube, silicon carbide fibers are taken, and then the target cell where you want to transfer your DNA, that is you want to make these cells as what the transformant, and the desired gene. Now you want to transfer this DNA into these cells. Naked DNA cannot be directly transferred into this target cells or the cones. In that case what is done, the silicon carbide fibers are used. When we use this silicon, that means in this test tube all these mixtures, DNA, that is desired gene, whiskers, and then the target cell into which you want to transfer your gene, all these are dissolved or they are taken in this test tube. When you do that, what happens, the DNA first it gets coated on the silicon carbide fibers. It gets coated over because it has got the higher affinity for the silicon, and then silicon with the coated DNA, easily what happens, it will adhere to the cells and get stably integrated with, first it gets integrated into the cell like this, the silicon fibers first they will integrate it into the cell wall and the cell membrane, then they will enter into the host, integrated into the host, and later they will attach themselves to the host genome. So this is how the genes can be transferred with the help of the silicon carbide fibers.